Okay, we're almost there, and we've got quite a number of people logging on, which is great to see. Again, for those that have logged on and you're just starting to sort of get used to this interface that you've got in front of you, in the top right, you've got a chat, a polls, and a handouts. The chat is obviously where you can potentially be having your own discussions, and ideally you would be putting your questions that we'll try and monitor and put to the speakers today. You've also got a polls tab that will intermittently be putting a series of poll questions that, if you've got 10 seconds, shouldn't take you much more than that to answer each of one of 4 poll questions that we'll be putting to you as the talks progress. And because of the sort of technical nature of these talks, we've also got a number of handouts in that top right sort of chat panel, which gives you things like application reports, papers, and relevant documentation that that both Kadla and Scott have been working towards. So a lot of the really great developments that they'll be discussing, they will be captured in some form in those handouts as various PDFs under that tab. So we're now at, just 4 minutes past the hour. I think at this point, I can probably bring Scott to the stage and we can start to introduce Scott. We've obviously got 2 speakers today, Scott Schachtele, who is product manager at FUJIFILM Cellular Dynamics, and we've got Kadla Røskva, who is my colleague at Sophion Bioscience and doing a lot of great work on these application developments towards recordings from iPSC-derived neurons that FUJIFILM Cellular Dynamics are world leaders in making. So just before I hand you over to Scott, I'll give you a very quick bio introduction to Scott. So Dr. Scott Schachtele is the product manager for the neural portfolio at FUJIFILM Cellular Dynamics. He works closely with research teams at FUJIFILM Cellular Dynamics to develop and deliver iPSC-derived neural cells for use in drug discovery and preclinical research. Scott received his PhD in neuroscience from the University of Iowa. Scott, that's enough of talking. If you can take the floor, and we'll look forward to your talk. Thank you.
All right, wonderful. I seem to be having some issues with my camera, unfortunately. So you may just have to not see my face, which may be good or bad. So at any rate, I'm going to share my presentation here. And surprisingly, it's starting at the very end. So we're going to go back to the beginning. A few technical hiccups. This is a live event, folks. All right, just as a reminder. Okay, so thank you very much for that introduction. And I'm very happy to be here speaking at the Sophion event on the development and characterization of human iPSC-derived induced excitatory neurons for disease modeling and drug discovery. So to start, just a brief background on FUJIFILM. So FUJIFILM has a legacy of innovation from film and more recently moving into healthcare, where they've acquired a portfolio of complementary life science companies to support preclinical research and discovery. The FUJIFILM Cellular Dynamics, or FCDI, is a part of this portfolio, and with it brings innovation in iPSC-based technologies, as well as industry-leading iPSC-derived cell manufacturing. FCDI has 4 core competencies that start with our proprietary reprogramming technology and licenses. And these fuel the flexibility of our platform due to the replicative ability of iPSCs to be a renewable source of starting material that can then be used and differentiated into many different cell types. As such, we have proprietary differentiation protocols that facilitate industry-scale manufacturing of a large variety of iPSC-derived cell types. And we are one of the oldest iPSC-based cell-based companies, and therefore we have unparalleled quality assurance and control around our products. So on this slide, I'm showing our off-the-shelf portfolio of iPSC-derived products derived from apparently healthy normal donors. As you can see, we have a number of cell types across various tissues and regions, including heart, liver, blood immune, and for the purposes of today's talk, looking at the nervous system, where we have a large portfolio of cells and offerings within that space. And these cells are delivered as cryopreserved vials alongside medium for culturing. And as I said, we're one of the, we really pride ourselves in our quality and our consistency and our scalability of our manufacturing. And so to demonstrate this and quickly illustrate it, this slide here contains purity data for 3 of our iCell neural products, our iCell GABA neurons, DOPA neurons, and motor neurons. As you can see, for each product, the purity is quite high, speaking to the specificity of our differentiation protocols. And also, you can appreciate the narrowness of the arrow bars, indicating the precision and consistency of our products. So this quality really translates into a reliable source of product for the researchers and research community, so you can focus on the questions you want to ask and not be thinking about deriving the models that you're going to use. We also have a wide portfolio of neurodegenerative and neurodevelopmental disease models, featuring a variety of genetically engineered and patient-derived models that include disease risk-associated mutations. And our portfolio focuses on 4 main areas, as you can see here: Parkinson's disease, Alzheimer's disease, neurodevelopmental disorders such as epilepsy, and then ALS and FTD, which will be a part of the talk here today. All right, to get started then, I wanted to first provide a brief overview and characterization of our iCell-induced excitatory neurons, and to start by really describing what I mean by what are induced excitatory neurons. So on this really basic illustration here on the slide is to depict a couple different ways that you can differentiate pluripotent stem cells into a neural phenotype. Or a neural cell. So on the bottom here in purple, one method is using directed differentiation, which guides pluripotent stem cells through key neural patterning pathways via small molecule treatment or growth factors or other morphogens that then lead it to become a neuron of specific regionality or specific cell type. On the other hand, on the top, we've got neural induction, and this method uses transcription-based approaches to differentiate iPSCs into neurons. So through the overexpression of transcription factors in the case of induced excitatory neurons, neurogene 2 overexpression, and then this can be done with and without growth factors or small molecules, depending on the protocol or the cell type that you're looking to derive. But some of the benefits of the model here of using neural induction or transcription-based approaches is that it can really help shorten the time to getting iPSC to neural differentiation. And it should work similarly across donors, which then relates to the scalability of the process working well across different donors. It produces a highly pure product that is functional. And then one thing is that if you're using transcription-based differentiation, this results in a non-regionalized neural cell type. So sort of a general glutamatergic or excitatory cell, as opposed to directed differentiation, where you can really specify regionality of that neuron in the central nervous system. So FCDI has manufactured NGN2 induced glutamatergic neurons and released them as iCell induced excitatory neurons. These come as fully differentiated, cryopreserved cells that are highly pure and designed for use in high throughput assays due to their low plating density and robust and reproducible performance. This next slide here shows the manufacturing process at a high level from iPSCs through NGN2 overexpression, maturation of the neurons, and then cryopreservation of them into what you would receive as a cryopreserved, fully differentiated glutamatergic neuron. One thing that FCDI specializes in is the optimizing product for downstream use. And for eye-cell excitatory neurons, we've done just this, building on the known scalability and purity of this differentiation method, and then controlling some of the known challenges in the system, such as control of impurities, doxycycline optimization for transcription factor, for turning on transcription factors, as well as adding additional manufacturing and quality control steps that can ensure the optimal product performance and reproducibility. And this is demonstrated in this slide, which is looking at our eye-cell-induced excitatory neurons, looking at purity in a number of different ways here. And so on the left, we're looking at flow cytometry for the 𝗵𝗿𝗃𝗃𝗲𝗿 tubulin, a marker of neurons, and this is using the 𝗃1 antibody. And we can show that we see really high pure neural cultures after coming out of this product, up in the upper 90% of cells staining positive for 𝗃1 across multiple lots. We've also looked at gene expression profiles across 4 lots shown here, and we can see that, again, these are very consistently expressing the same markers across the different lots, and this includes glutamatergic markers like AMPA receptor subunits, the 𝗲-glute receptor transporters, as well as synaptic markers like PSD95 and synapsin1. And we can stain for some of those markers as shown in the ICC image on the right-hand side. Both our healthy and our disease cells can be cultured in a variety of permutations, from cells in monoculture to multicellular cultures in 2D or 3D. And this slide illustrates this and lists some of the more common assays used under each situation. The point being that you can really utilize our cells and tailor your experiments to answer specific biology of cells or of the disease of interest. So for our induced excitatory neurons, one common characterization and testing method we like to use is microelectrode array, or MEA technology, which allows for the network level recording of neuronal signaling. The applications team at FCDI has put a lot of work into understanding MEA technology, and we provide protocols for culturing our neurons on this platform, and these can be downloaded at the FCDI website. In the set of data shown on this slide, we're using the Axion Maestro MEA platform and co-culturing induced excitatory neurons with our eye-cell astrocytes 2.0. So when we do MEA culturing, we encourage neurons to be co-cultured with astrocytes, as the astrocytes help mature and develop the synchronized bursting of neural networks and really mature the network formation. And this can be observed in the example data shown on this slide with these roster plots on the top, showing that we can see spontaneous activity showing as early as 3 days in culture with the individual tick marks on the roster plot, and then that these mature into nice synchronized bursting patterns by day 21 and 28, symbolized by the high level peaks that are shown on the top of the rosters at those time points. And within the software, we can analyze a number of MEA parameters like total activity, synchrony, burst strength, to help quantify network development. And we also, in data not shown here, but we've also used the MEA to interrogate cell function using known neuromodulators such as GABA, AMPA, and NMDA agonists. Again, speaking to the consistency of our manufacturing for the eye-cell excitatory neurons, we can look at MEA activity across multiple lots of differentiated product, and we observe consistent formation of synchronized networks across these lots, as you can see from the 3 different roster plots on the screen. So following our protocols and using our cells, very robust assays can be set up with the excitatory neurons. And then last, we've also established that our induced excitatory neurons can be cultured in 3D as eye-cell neurospheres. And we have a protocol on our website that instructs how to form these neurospheres. But sort of simply put, you can just thaw our eye-cell induced excitatory neurons, as well as our eye-cell astrocytes 2.0, and combine them in a suspension at defined ratios, and then plate these at approximately 25,000 cells, and from that mixture into a ULA round bottom 384 or 96 well plate. And then the cells will form neurospheres within 2 to 3 days. And network activity can then be monitored by calcium imaging, which, as you can see as on these oscillation plots shown at the top of the slide. And this is a nice method for utilizing induced excitatory neurons in a high throughput 3D culture workflow. And again, the function of these neurons can be validated through the addition of neuromodulators, much like what I just mentioned with the MEA platform. Okay, now I'd like to move into discussing how we're using our induced excitatory neurons as a model for frontotemporal dementia using our granulin 𝗵𝗿𝗰𝗏𝗏𝗃𝗏 heterozygous knockout neurons. So frontotemporal dementia and amyotrophic lateral sclerosis, or ALS, are on a disease spectrum with common underlying gene mutations that can either lead to motor neuron disease and cortical degeneration and subsequent or cortical degeneration and subsequent behavioral changes. Within this spectrum, mutations in the progranulin gene, circled here on the screen, are a predominant risk-associated mutation for frontotemporal dementia, with up to 20% of familial FTD and 5% of sporadic FTD attributed to it. And specifically, GRN mutations, such as the 𝗵𝗿𝗰𝗏𝗏𝗃𝗏 missense mutation, result in a haploinsufficiency of progranulin in the brain. And some of these implications for this granulin deficiency include protein mislocalization, lysosomal dysfunction, neuronal and synaptic loss. So research models for FTD and granulin deficiency have been in large part performed in mouse knockout models, where a full homozygous knockout is typically used to manifest these disease-relevant phenotypes. More human-relevant models are desired to help improve the efficiency of drug discovery and translation, and so more recently, human iPSC-based models are being employed. And so as such, we've generated induced excitatory neurons featuring a granulin 𝗵𝗿𝗰𝗏𝗏𝗃𝗏 heterozygous knockout. And we've confirmed that these have reduced granulin compared to the healthy control-induced excitatory neurons, and this is shown on the left-hand side of the slide, and looked at using simple Western. And we also know that the differentiation process for the mutant line is robust, producing similar purity compared to the healthy control and across multiple lots. And this data is shown as 𝗵3 tubulin percentage across multiple lots here on the right-hand side of the slide. All right, so as mentioned earlier, formation of neural networks on multi-electrode array is a great way to monitor neural network development and identify disease phenotypes. From mouse studies, we know that there's a significant brain pathology associated with the GRN mutation. However, there's not much known about the effects of this mutation on neural networks. A few recent papers have been published with mixed results showing little to no reduction in MEA activity on the left, to potentially increased activity shown in the data on the right. Both these studies here use iPSC-derived neurons with a homozygous knockout for GRN, which isn't typically what you manifests in the clinical setting. So what we wanted to do with our induced excitatory neurons was examine that using our heterozygous, our GRN 𝗵𝗿𝗰𝗏𝗏𝗃𝗏 heterozygous knockout. And so using our induced excitatory neurons on MEA, our initial results shown here show relatively similar rates of network development between the healthy and the FTD neurons. In this representative plots, the GRN knockout does look to have increased synchronous firing, but this effect is pretty slight, but it is consistent. A more obvious differentiator is shown on the right side, where we look at more acutely at the dynamics of the peak bursts within these networks. And in doing so, we can see a very reproducible phenotype where the GRN knockout neurons have a slower burst time compared to the healthy. And these data suggest that the granulin neurons may have a slow depolarization rate compared to apparently healthy normal neurons, or some other effect of the network development. So we recently collaborated on an application note with Maxwell Biosystems that have an HDMEA 24-well plate system. This allows for high definition recording of neural activity, along with, and similar to the 𝗂𝗄𝗏 Maestro, the ability to monitor activity longitudinally. So in this collaboration, we compared the healthy controls and the granulin knockout line. And you can see just representative activity plots side by side here between the healthy and the knockout. And you can see qualitatively increased activity in the bottom right-hand corner of the disease line. And we can, quantify that and plot it. And we see that comparing the GRN to healthy controls, the GRN shows overall higher firing rates and a large percent area of activity compared to the healthy control, suggesting the cells are more active. This is similar to what we saw with the Axon system, but the differences are maybe a little bit larger on this platform and more accentuated at the longer time points that we looked at. We also compared time to burst peak on the Maxwell system, shown on the right, similar to what we did on the Axon. And we do see that the longer burst peak timing that was seen on the Axon system is also reproduced on the Maxwell. So the Maxwell system also has a module for axon tracking, where we can isolate the origin of neural activity and track along the well. This video on the right is static right now, but we can see this on the images at different time points on the right here, looking at axon tracking across multiple time points. One thing you can appreciate is that if we follow the same neuron in culture over time, we see an increase in the complexity of the axon tracking events, which are indicated by the red lines, increasing complexity of the red lines marking the neural tracking events. And these suggest continued network maturation and connectivity over time in the culture. And comparing the GRN R493X to healthy controls using the axon tracking software, we can see consistently longer, more complex connectivity in the healthy controls compared to the disease model. This is also supported, this is shown on these example images on the left-hand side, but is also supported on the right with a neurite outgrowth assay recorded on the INCU site, which also shows longer neurites in the healthy controls compared to the disease line. So to put it all together, we're observing that while the granulin R493X induced excitatory neurons have higher firing frequency and levels of activity, they're more limited in their connectivity and maturation of their neurite network. One theory is that the higher activity is resulting in emerging excitotoxicity in the GRN model that could be manifesting as and truncating the network development. But further experimentation is needed to understand that. And then on the last slide here, in summary, we're really in early days of understanding how these MEA data correlate to dementia and frontotemporal dementia. But I hope that I demonstrated that these iPSC-derived excitatory neurons and the FTD line are a robust model for modeling neural function and neurodegeneration. And with that, I'll just say thank you for listening, and I'll pass it back.
Great, Scott. Thank you. That was a lovely talk. And I'm sure we'll have plenty of questions at the end for you as well. So if you can just return for the last 10, 15 minutes or so of this webinar, we'll put those questions to you and Kadla in due course. And now on to our second speaker today. So this is Kadla Røskva, my colleague at Sophion Bioscience, who will be talking around her characterization via automated patch clamp of some of these FUJIFILM Cellular Dynamics iPSC-derived neurons. So to give you Kadla's bio, Dr. Kadla Røskva completed her PhD in 2014 at Copenhagen University using live cell fluorescence microscopy to quantify the localization and function of membrane proteins. In a subsequent postdoc at the 𝗵𝗿𝗁𝗏 Cardiac Research Institute in Sydney, Australia, she established an assay for quantifying the structure-activity relationship of ion channels using a combination of high-resolution microscopy and electrophysiology. since 2017. She has worked as a research scientist at Sophion. I believe she's even higher than that now. I think she must be a senior research scientist. So this bio is putting you down, Kadla. I will tear this up and burn it in honor of your higher level. She's a senior research scientist at Sophion, heading a program to enable high-throughput automated patch clamp measurements of iPSC-derived cell lines. Over to you, Kadla.
Thank you so much, Damien. And I also had to run my presentation all the way to the back, but now it's ready. So I'm a scientist working at Sophion, and today I will present a project where we used automated patch clamp to evaluate disease phenotypes in iPSC-derived neurons. As Scott already mentioned, induced pluripotent stem cells are produced from somatic cells that you can extract from a patient. They are then reprogrammed into stem cells that you can differentiate and mature into the specialized cell of interest. In this case, we're working with iPSC-derived neurons. Advantages of iPSC-derived neurons as disease models, I have written down in the yellow box below. They are more accessible than primary human neurons. They're more translational than animal models. They can capture late onset diseases and have an ethical advantage as compared to embryonic stem cells. So this technology has given access to many different neurological disease models, but that has also created a requirement for high throughput platforms for their characterization, disease modeling, and drug screening. So in automated patch clamp, you obtain high throughput single cell measurements of ion channel currents and action potentials. These are measures that are very relevant to evaluate neuron function. And for that reason, we wanted in this project to establish the methods required for using automated patch clamp to evaluate disease phenotypes in iPSC-derived neurons. As a proof of concept disease model, we worked with a frontotemporal dementia FTD cell line that was kindly provided by FUJIFILM Cellular Dynamics. Scott already introduced it, but as a short recap, in about 20% of genetically inherited FTD, the disease is caused by mutations in the GIN gene, which causes lower levels of the growth factor progranulin that is involved in neuron development, function, and maintenance. This has a lot of downstream effects and can lead to cellular pathologies such as lysosomal dysfunction and neuroinflammation, and eventually neuron degeneration and death. So FCDI produced excitatory neurons carrying one of the most abundant mutations in the GIN gene. And they showed that this population of neurons have reduced protein expression of granulin as compared to the wild type. So these are the two neuronal populations that we're working with in this project. Then, when you want to record neurons with automated patch clamp, a very crucial step is that you have to dissociate the neuronal network into a pure and viable and homogeneous cell suspension. This process is challenging and it often requires optimization. On the left, you can see both on the culturing stage, you can work with plating densities, different surface treatments, you can add stabilizing enzymes. During the dissociation, there are different types of dissociation enzymes. You can vary incubation times and centrifugation conditions. You can also replate the cells. And then the neuron suspension will often require some sort of purification after they have been dissociated, either with filtering or sorting using a gentle cell sorter. So for the neuron models that we have worked with, we have made operating procedures for the dissociation that are available upon request. And we have also just released a step-by-step guide as to how you can optimize the dissociation in your own laboratory with your own iPSC-derived neurons. Once you have a good cell suspension, you can add it to the automated PET/CLAMP instrument of choice. In this case, we worked with 𝗃𝗃𝗃𝗃 and 𝗃𝗃. On the 𝗃𝗃s, the minimum requirement, the minimum number of cells required for a full consumable with 48 experiment sites is 200,000. And on the 𝗃, the minimum number of cells required for a consumable with 384 sites is 2.7 million cells. And I think on our newest technology, the 𝗃3, this has been lowered down to 2 million cells. If you need to go down in cell numbers even further, which is sometimes the case with neurons, you can also work with partial consumable plates. And there's also the possibility to test multiple cell clones in parallel, which is really an advantage if you want to do a side-by-side evaluation of a disease model neuron in comparison to wild-type neurons. So here I show a consumable plate view of the Q-chip, which is the consumable we use on 𝗃. It has 384 sites, and each site harbors a single cell. Then we have filtered the data based on some filters on the membrane parameters, the membrane resistance, and the cell capacitance. And with those filters, we obtain a success rate about 60%, as color-coded here in green. Then on each site, you can run subsequent experiment protocols. Here we have recorded voltage-gated sodium and potassium channels. Then we have used current lamp to record action potentials. And then we have recorded ampere-mediated currents in the end. So already at this stage, you can get an idea of the electrophysiological properties of the cells. So here on the right, I have plotted the percentage of the neurons in which we could record voltage-gated sodium and potassium currents, the ampere-mediated currents, and action potentials. And we recorded this at day 14 in culture and day 35 in culture. And what you can already see is that the cells or the neurons are quite variable in terms of their electrophysiological properties, and that the properties are changing over time. For example, the percentage of neurons that are able to fire action potentials increases quite drastically from day 14 to day 35. So then when you want to compare your disease model to the wild type, it is important that you aim at comparing neurons that are of similar functional maturity. So to do that, we first did a more in-depth pharmacological and biophysical evaluation of the voltage-gated sodium and potassium channels. I'm not going so much into details with this data. I just want to tell you that we use this to select the population of neurons that were expressing these channels. And then we use those, or we further classified those based on their action potential firing pattern. So we did a very simple classification into 4 types. The type 1 neurons were not able to fire action potentials. Type 2 neurons could fire a single action potential. Type 3 neurons fired 2, and type 4 neurons were able to fire more than 2 action potentials. And then we could plot the proportion of neurons falling into these 4 categories as a function of maturation time here in the bottom left corner. And both on 𝗃 and 𝗃, we saw the same trend in the data, that the proportion of the neurons that were more functionally mature increased over time. We can also look at the membrane or the cell parameters that are recorded in the different neuron types. So in the graph in the middle, you can see that the more mature neurons are slightly larger. They have a slightly higher capacitance, and as might be expected on the right, that they have a lower resting potential. Then when we look into the ion channel measurements that we can do in the different neuron types, we can see that the more mature neurons have a significantly higher sodium current density and a slightly lower steady state potassium current density as compared to the less mature neurons. And when we look at the AMPA-mediated currents, we can see that we can record these currents in a higher proportion of the more mature neurons. So this is all data recorded in the wild-type neurons. Now we can do the same analysis on the disease cell line, and we can start comparing those. So again, both on 𝗃 and 𝗃, we found that the proportion of the type 4 more functionally mature neurons were lower as compared to wild type, and we did not see this increase over time. Then we compared the type 4 neurons, and we saw that the disease neurons were significantly larger than the wild type. We found a small, non-significant trend in the data towards the disease cell line having smaller potassium currents as compared to the wild type. And then we saw that the disease neurons had significantly higher AMPA current density and significantly lower sodium current density as compared to the wild type neurons. So now we can try and see if we can relate these observations on the ion channel currents to the neurons' action potential firing. So we looked at the current clamp protocol we were executing. So we used a current step protocol, and we analyzed the action potentials in response to the current step that elicited the highest amount of action potential peaks. And we could quantify that the disease cell line had a significantly lower peak potential and a significantly lower action potential amplitude and a lower depolarization rate as compared to the wild type, which is in good agreement with a lower sodium current density. So now, of course, it's also relevant to start looking at how our data compare to network behavior. And very preliminary observations are that from the data that FCDI has, is that the network has shorter and less complex neuroid outgrowths as compared to the wild type, which indicate that that the neurons are slightly less mature, which is in agreement with what we see, that the proportion of the more mature neurons is lower in the disease cell line. And also they do quantify this longer time to peak in the disease cell line in agreement, or at least in alignment, with a lower depolarization rate and lower sodium current density that we observe. So to summarize the project, by optimizing the dissociation, we can use automated PET clamp to evaluate neurological disease models. We find that the technology is useful for identifying neuronal subpopulations based on ion channel currents and action potential parameters. And using this strategy, we observe that the disease model we are working with contain a lower proportion of functionally mature neurons as compared to the wild type. And we can reveal or quantify differences in ion channel currents that correlated with action potential properties. So now, here in the end, I just want to spend 3 minutes mentioning some ongoing developments that might be relevant for neuron applications. First of all, on the left here, we have some very preliminary data where we are looking into modulation of neuron excitability by the addition of compounds. So on the top, you see hyperpolarization of a neuron by addition of 𝗵𝗂𝗃𝗏𝗏. Again, data is scarce and it's preliminary, but there's a trend in the data towards that additions of 𝗵𝗂𝗃𝗏𝗏 lowers the resting membrane potential and increases the wheel-based current. On the bottom, there's an example of depolarization of a neuron by the addition of 𝗵𝗂𝗃𝗏. So in black, you can see the response of a neuron to a current ramp. The membrane potential increases until it hits a threshold and the neuron starts firing action potentials. Then upon the addition of 𝗵𝗂𝗃𝗏 in red, the membrane potential is depolarized above the threshold, and we don't see a response from the neuron. And then after addition of 𝗵𝗂𝗃 and a blocker of 𝗵𝗂𝗃 in gray, the membrane potential is restored and the cell can fire action potentials again. Another thing that we have been working on is a feature for the adaptive recordings of the wheel-based current. So this feature allows us to, within a single experiment, record the wheel-based current and apply it in subsequent protocols. We already have a beta version out, and with the preliminary data we have on this, we can see that this helps us overcome the cell-cell variability that we see when measuring action potentials and helps making the data more reproducible. Then we are also working on a perforated patch of neurons as an alternative to the standard whole cell suction. So in very brief, in this strategy, you use ionophores that insert into the membrane and provide electrical access to the interior of the neuron without compromising the integrity of the intracellular environment. And we believe this strategy might provide more physiologically relevant measurements. And finally, we also have an ongoing collaboration with Nottingham University, where an industrial postdoc is exploring APC of network neurons. So with that, I just want to acknowledge the people at Sophion that was involved in this project, our collaborator, Scott. I want to mention that we have a lot of experience now with primary and iPSC-derived neurons. If you think it's interesting, we are always open for collaborations. And we also have Sophion travel and research grants that are available if you are an early researcher. Thank you for your attention.
And I guess if we can get Scott to join us, we can potentially use the last 14 minutes or so of the hour that we've allocated for plenty of questions. Both you and Scott Schachtele have your work cut out because we've got quite a lot to get through. I will start with the first one answered, which is if you are using the question mark tab, the people that will be facing the question, Scott and Kadla, this is at the very bottom of your question mark, as in where the questions are. So Ilya asked quite early on, and this will be directed to Scott, what is the degree of automation for iNeuro manufacturing?
Yeah, there we go. Okay. Good question. We use proprietary manufacturing methods to be able to do all this, but the idea is to make it really reproducible and optimized and scalable. And so, we can't comment on the degree of automation or automation that's been done on it, but just know that we go through a lot of quality control and process to be able to produce a reproducible product.
And another one for you, Scott, while you're on. Bob asks, how do icell-induced excitatory neurons differ from icell gluteneurons?
Sure, yeah, good question. So our icell gluteneurons, I didn't explicitly talk about in this, but that's another product that we have. Those are the differences is that those neurons are differentiated using the directed differentiation approach. So meaning they're going through neural patterning pathways and being specified to become cortical glutamatergic neurons. The induced excitatory neurons are glutamatergic as well, but they're using neural induction or transcription-based approach, which is forcing them into the neural becoming neurons. And those, while having lots of features of glutamatergic neurons, they're not regionally specified, meaning that we do see some markers of like peripheral markers like perforin and other things in there. But in all intents and purposes, they do have a lot of the same channels, receptors, and functions as cortical glutamatergic neurons. So in the way that we think about it is that the induced excitatory neurons are really well suited for high throughput, high level preclinical screening. Once compounds are defined or refined to a smaller list, this could be then moved into a more cortical glutamatergic product with our icell gluteneurons.
Thank you. This one is presumably to you, Kadla. So Ilya has a second question. 60% in terms of your success rate seems wasteful. What can bring the success rate to maybe 75% at least?
Yeah, so the success rates are always different from cell line to cell line. So for example, the gluteneurons that Scott just mentioned, they run at about 75% success rates. So there's always that factor that every iPSC-derived cell line that we work with has different challenges and different success rates. In general, if you have more cells, so you can work at higher density, that tends to increase the success rate. These, when you work with iPSC neurons, the number of cells tends to be a bit scarce, and that hampers success rates. And then the purity of the cell suspension. So sometimes going through a more extensive purification, for example, using cell sorting, can also increase the success rates. But I think in the very best case with the neurons, that is around 70–75% that I have seen so far.
And whilst on success rates, Kadla, I'll just skip a question. We will come back to this, so don't worry. I'll try and get as many as I can. But Chris asks, can you better define the success rate criteria? How long were the experiments? For example, any compounds tested as part of maybe that success criteria?
Yeah, so, well, the basic the success rate, the QC success rate that I mentioned in the presentation was based on those membrane parameters, and those were throughout the experiments that were used in this analysis. So they were probably, how long were the experiments? I think they would probably be around an hour. And yes, we also tested compounds. So it was kind of a simplified version I showed. We tend to run experiments as you saw, voltage gated, then current clamp, and then the ligand gated, and then we would run voltage gated in the presence of compounds, or we would run current clamp in the presence of the ligand that we add, or something like that. Yeah, I hope that answered the question.
Thank you.
Kinda.
And then another one for you, Kadla, which is one of the questions I skipped to take Chris's while we were on that success criteria style question. So Adele asks, have you tested iPSC-derived sensory neurons for mechanosensitive or nociceptive ion channels?
I don't think that we have. We have an ongoing project with red blood cells where we test for mechanosensitive ion channels, but with the iPSC-derived neurons, I don't think so.
Can I maybe jump in here too to add a little bit? Yeah, so we do have our eye cell sensory neurons, which are iPSC-derived sensory neurons, and we've done quite a bit of characterization of them, kind of outside electrophysiology, mostly testing for function of mechanosensation or nociception using calcium assays, and we can add those agonists for mechanosensation and nociceptors, and we see increased calcium. So we have done a little bit of that on our cells, and happy to share that with you, Adele, if you want to connect with me after the webinar.
And I'll quickly jump in while you're still around on the line, Scott, and ask you something around the genotype. So you showed some nice genetic sort of screening data almost for some of your iPSC-derived FTD neurons. How do you find that RNA sequencing, that genotyping almost of your FTD neurons, or maybe even some of your other disease neurons? How does that compare to maybe what the literature suggests, and you know, and in terms of how that genotyping might then relate to, you know, the disease state phenotype that you might be looking for?
Yeah, sure. Great question. Thanks. So I can speak to, what we know for our induced excitatory neurons on this, maybe first and foremost. So as I showed, as part of the characterization, we have done some RNA expression comparisons between our apparently healthy normal and our FTDs. And we do see very similar expression of things like synaptic markers, synapsin-1, PSD95, key glutamatergic and neuronal markers like 𝗵𝗿3 tubulin, vGlut1 and 2, AMPA receptor subunits. In terms of sodium channels and ion channels, that would affect neural network firing and connectivity, and maybe some of the things that Kadla was observing. We don't see at the gene expression level much of a difference in sodium channel expression or potassium channel expression. But again, we did look at relatively early at our RNA data is from an earlier time point, so around day 16. So it's possible that at that stage of development, that later stages of culture, that some differences may be observed at day 20 or day 30, as what was looked at with some of the patch clamp data. So, I mean, that's one thought. On the other hand, phenotype differences that we observed in the induced excitatory neurons could be more at the protein level rather than the gene level, where it's localized, how much is being translated. But this is a great question. I think there's a lot of experiments that can be done to help try to tie those couple of things together.
Thanks, Scott. So going back to the list, and we're trying to get through as many as we can. We'll try and get to the end of these before the end of the hour. We might stray a minute or two over, but we probably won't be holding you too much longer. So Esteban asks, or even comments at least initially, thank you for the nice presentation. This is probably for Kadla, actually. Just a naive question. Since the vial contains up to 6 million cells, have you tried placing the excitatory cells directly from the vial into the cube instead of culturing them, or is the culturing step necessary for cell maturation, for instance?
Yeah, so with these cells, we haven't tried it, and in general, the culturing is required for their maturation. But we have actually previously tried it with iPSC-derived neurons, and it actually works. You can get them onto the cube and they patch, but they do have a very immature phenotype at that point and very small currents.
Okay, so moving on, and I think this might also be for you, Kadla. I think this is the audience we've got, so not sort of be put off, Scott. I think we've actually got a lot of APC ion channel aficionados out there. So a lot of these are heading to Kadla. Kadla, Jean, who I believe might be Patrick as well, have you recorded HCN currents from from these neurons, sorry?
No, we haven't tried that. Not for these neurons, no.
Okay, but something to try, because you know there's a big community of HCN people out there, me included. Okay, moving on. Iliada asks, do these neurons have spontaneous action potentials? And I guess that could be to both of you actually, because I think Scott, you will have done some of this with your MEA recordings too.
Yeah, I can quickly answer for the dissociated neurons that we work with. In general, they don't. Not necessarily because they wouldn't do it in a network, but I think this whole dissociation process is affecting them somehow. And we do see it rarely, but in general, not. I think it's different in the MEA.
Yeah, to jump in there, we do see spontaneous activity on the MEA. And this may be, we start to see early on, small on the MEA, it's recording from an area within the network. So it's not down to the single cell resolution, but we do see within those like small uprise in spontaneous generated activity within them. Now these could be driven by the early formation of synaptic connections between the two, so that maybe not purely spontaneous out of an individual neuron, but provoked by initial connections locally between individual neurons that then sort of expand over time in culture. But yeah, we can see small activity to start.
And then Alison asks, how long can you culture or compound treat neurons on a chip before analysis, or are they plated immediately before patching and you'll lose most projections in the dissociation and seeding? And again, obviously that could be, in terms of the APC characterization, but I guess Scott could even potentially answer some of that around where they're not dissociated in MEA recordings. But maybe you start first, Kadla.
Yeah, so for our experiments, the cells are dissociated and they have retracted their protrusions. I think there has been a bit of experimenting around culturing cells on the chip, but so far we cannot do that. So we work with the cells dissociated and they are sucked onto the patch hole. So they will be depleted of their protrusions.
Yeah, I can just add, from a monoculture or a co-culture perspective, these can be cultured for extended amounts of time, 4 or 5, 6 weeks. With respect to the dissociation, Kadla mentioned that it can be gets more complicated as the cells age because the networks and the projections get more complicated. So it can be hard to retrieve single cells, but yeah, you can culture them for multiple weeks in monoculture.
Thank you. And we are over the hour mark now. We can maybe take another 2-3 questions, but at some point we probably will need to stop, and obviously a number of you will need to be getting on with your working day or whatever you might be doing. But we'll try and take a couple more and see how we go getting on. So Bob asks, for current clamp recordings, again, this will be more, I guess, towards Kadla. what percentage of neurons have negative resting membrane potentials?
I think the majority have negative resting membrane potentials, but they are slightly or they are quite depolarized in general when we work with dissociated iPSC-derived neurons. I think the excitatory neurons are doing very well on that matter, and their resting potential will be somewhere between minus 70 and up to minus 20 millivolts, something like that.
And Lars asks, have you tried measuring the neurons using cesium fluoride in your intracellular solution to isolate sodium currents?
Yes, we have. We actually do that as part of some of the experiments. So we use different intracellular solutions across the plates. Some of them with potassium in there, and some of them with cesium.
And Sumin asks a slightly more involved question. Was the change in firing properties of the granulin disease model controlled for the apparent depolarization, reduction of the minimum voltage trajectory of the action potential? Or in other words, is there some sodium inactivation that is occurring that could explain the difference?
That is a good question. I think I would have to look closer into the data to explain that. We do see a slightly offset, a small offset in the activation curve at the very low voltages, with the disease cell line being opening a bit earlier than the wild type, and that we have had some speculation could affect this, but otherwise I think this is something I would have to come back at when I had a closer look at the data. Yeah. And
we're now 4 minutes over the hour, but we have 2 questions left. I'm going to try and answer them or ask them both. So Enoch asks, which system or platform do you recommend for gentle sorting? In my experience, sorting severely compromises cell integrity and reduces our success rate on the 𝗵𝗄384 compared to unsorted cells.
We are working with a Miltenyi cell sorter, and that has proven very good at producing cells that we can straight after put on our systems. We can even keep them for a while, maybe for an hour or 2 in the fridge, and then put them on, and they are still doing relatively good. So that would be my recommendation.
And Enoch, if you need any information on that Miltenyi cell sorter, just get in contact, because we've been using it in our labs for probably 2 or 3 years now, and it's probably, as Kadla suggests, one of the most gentle, and yet it does a good job of sorting without, as you say, these compromising cell sorting issues that you can face with less gentle cell sorting. Right, final question. So we're going to get through it, hopefully. And Dylan asked the question. Last question of the day is obviously the best question of the day. So no pressure, Dylan, but this better be a good one, because we've waited around for nearly 6 or 7 minutes after the hour. Thank you for the presentations. Regarding the automated patch clamp, has any characterization been performed to compare the function of cultured neurons attached to a cover slip with dissociated neurons on the automated patch clamp?
That is a good question. I think we actually did this comparison, but it's quite some years ago, and I think it was published. I think if you reach out to us, I can find that paper, but otherwise you are right. For many of these things, it's only recently that we have really started getting good enough success rates that we can actually get very useful data out of our measurements with neurons. And it's extremely relevant to see and compare how our data compare to neurons that are attached to the surface. So this is not something that we have done recently, and it would be super interesting to revisit if we get the right collaboration around that.
Great. That just leaves me to wrap up now. I want to thank Dr. Scott Schachtele and Dr. Kadla Røskva for their excellent talks, and I think the really interesting Q&A at the end there as well, with a lot of input from you all in the audience. So thanks to you, the audience, for your attention and your time, and some really interesting questions that hopefully we've answered them as we've been suggesting throughout. If you want further questions, you want further information, maybe you want some of those papers or PDFs that we've been suggesting may help answering some of your questions further, do feel free to reach out to us. I think you've got an email, info@sophion.com would be a standard one that you could direct it to. Otherwise, 𝗵𝗵𝗿𝗁, I'm very active on LinkedIn, so you can always catch me there. And I'm sure we've got plenty of other ways that you can get in contact with us should you have more information. And of course, any of these questions that are specifically directed to the experts, we'll forward on and hopefully get their feedback for you in due course. So thanks to you all, and thanks to our speakers once more. Take care, everyone, and enjoy the rest of your day.